In this video we will discuss about HIV immune response, that is human leukocyte antigen response. So let us start how the HIV virus infect the cells. So it infect the cells that have CD4 plus and CCR5 receptors, and when it's uh, after endocytosis, viral RNA is released, and with the help of reverse transcription, it is converted into DNA, and then it is integrated to the host genome. Then the translation process occurs, uh, which is polyprotein translation, and the protease that is that comes with the virus helps to cleave them in the form that it can assemble and uh, proceed towards budding, and then it releases from the cell, and then can infect the other cells. So this is the life cycle of how the HIV virus infect the cells and produce more virus to infect other cells. However, the other thing that people have identified is how the immune response get activated. So some of this polyprotein, after protease cleavage, gets to the proteasome, where it is converted into, it is been digested or form into peptides that binds to the HLA class, that is human leukocyte antigen class, and this happens in the Golgi, and from the Golgi it will then transported to outside the cell or on the cell surface, uh, that can triggers the CTL, that is cytotoxic uh, cytotoxicity T uh, cytotoxicity T uh, receptors, or this is T cell receptors, and this is immune response. I am not able to recall the full form of CTL. You can get this full form from the reference that has been provided below. So let us continue that there are three kinds of HLA expressions are occurred. One is HLA-A, another one is HLA-C, and third one is HLA-B 5701, and these are combined together called as HLA class 1. So they will then form a receptor at the on the infected virus that is recognized by yeah, T cells, and then it can trigger a immune response. So how these different kind of expressions leads to different kinds of response, we will discuss that in next slide. So this is the slide where it shows how different kind of HLA will affect the immune response. So when the HLA-A, there are two kinds of HLA-B allies. Uh, One is protective and another one is non-protective. The protective HLA-B allies will be able to produce more peptides of different epitopes on the cell surface that can result in polyfunctional CTL recognition. That is, it will train the immune system better, as the immune system will recognize different epitopes of the virus and will be able to perform its function better. Whereas when the HLA-B is not protective, then it will not be able to produce many kind of epitopes, and the response will be limited towards a few, against a few epitopes that are presented on the cell surface, as it can be seen. And this, in this figure, blue is represented as monofunctional CTL recognizing, that is less diverse, and this green is considered as polyfunctional CTL recognizing. Here it is described uh, for your reference. And second one is HLC allies. So HLA-C allies. So what happens in HLA-C allies is that uh, the mRNA that is required to form HLA-C allies uh, sometimes absent, and sometimes they are present. The 3' ends sometimes not able to bind. In this case, when the 3' is not able to bind the mRNA, then HIV virus will not be able to suppress this HLA-C allies. In this case, more receptors will be, more epitopes or are being available on the infected cells, whereas when the binding site is available for this HLC allies at 3' end, this HIV virus will be able to suppress this mRNA, this HLA-C allies through the binding, and in this case we will not be able to see too much uh, epitopes on the cell surface compared to the previous case where 3' end is not able to bind. 3' end of miRNA, microRNA of this HLA-C is not able to bind. So in this case HIV will not be able to suppress it, whereas in this case HIV will be able to suppress it. The third one is about HLA-A. So when the HLA is expressed at low amount, then it will not interfere with the natural killer cells, and it will help the natural killer cells to recognize this HIV target cells. However, when the HLA is expressing at a high amount, it will affect the response of the receptor that is present in natural killer cell, that is NKG2A, and, and it will prevent this receptor to bind with HIV cell virus. So in high HLA expressing condition, natural killer cell will not be able to perform its function uh, as it is required, or as compared to where HLA is expressing uh, in low amount. So this is how the different expression of HLAs lead to different kind of response in HIV. That's all for this video. Thank you for your time.